building immune resilience and active aging, epigenetic strategies for revitalizing aged immunity and revising immune paralysis in sepsis.
Thank you, and thank you to the organizers. It's been a pleasure so far. I've really enjoyed myself, and I'm going to be very happy to share some of the information and data that we've been generating in the university and the Mater Dei Hospital related to improving resilience in the immune system as we age. So globally, people are living longer. This is inescapable now. By 2030, 1 out of 6 people in the world will be aged 60 years or older. And with progressive aging come various health challenges, namely cardiovascular diseases, neurodegenerative diseases, and in particular infections, pneumonias, abdominal infections, skin infections. And these infections can lead to severe complications, for example, sepsis. A few numbers here. Adults aged 65 years and older are 13 times more likely to be hospitalized with sepsis than adults younger than 65. In addition, around 63% of adults over 60 years are admitted to the ICU presenting with sepsis upon admission. But what is sepsis? It's currently defined as the dysfunctional immune response to an infection leading to lethal organ dysfunction. It is estimated that there are yearly 48.9 million incidences of cases of sepsis worldwide, with around 11 million deaths. And this far outweighs all deaths caused by cancer cumulatively. So sepsis is a leading cause of mortality and morbidity worldwide. All major organs can be impacted by sepsis. And this can be caused by various infections due to respiratory microbes, for example, Haemophilus, Staph aureus, Streptococci, and also SARS-CoV-2, the causal determinant of COVID-19. In addition, we have E. coli and more gram-negative bacteria together with fungi that can cause abdominal sepsis, or most often referred to as fecal peritonitis. The respiratory system is by far the most prevalent primary site of infection. To date, despite these alarming numbers and a World Health Organization resolution in 2017, no specific treatment for sepsis has yet been approved. Despite over 150 clinical trials targeting the inflammatory cascade, which we suspected was the core, the root of the problem in septic patients, were unsuccessful. So this called for a reevaluation of our model systems and pathophysiology of sepsis. To complicate matters, indeed there are those who survive sepsis. So there's a large survivor pool. And these survivors continue to suffer long-term complications. For example, fluid retention, cognitive decline, persistent inflammation, and immune dysfunction. Nowadays, we know this due to the sensationalization of the media as now long COVID. Well, long COVID, COVID-19 is a viral sepsis. So we've known about post-acute infection syndromes and long-term sequelae for quite some time. And patients, unfortunately, who survive sepsis are at high risk of readmission. So what have we done in the past? This was a study in 2017 where we tested the new and up-and-coming technology related to genomics. So we've looked at a diverse septic patient population. This is a population in the Netherlands. We've obtained blood samples on ICU admission from 522 patients. We've mapped the transcriptome of these blood leukocytes. And what we found was that we can stratify patients into 4 groups. Here we named them Mars 1 to Mars 4. And this is solely based on gene activity. While clustering in itself is neat, and we can actually cluster people around here, that's one thing. However, it's another thing when we have a clinical implication. And in fact, what we found was that MARS1 classified patients were at consistently high risk of mortality, starting from 14 days up to 1 year. Besides that, we also looked at the pathways where these genes are functioning. And what we observed was that the MARS1, or the poor prognosis endotype, as we're calling them, was associated, characterized by immune dysfunction, or what we're coining as immune paralyses. So we have now a tool whereby we can identify patients who are characterized by an immune suppressive state, and this may actually lead, we hope, this may lead to a more precise treatment regimen, where in this case, MARS1 patients being immune suppressed will not benefit from, for example, corticosteroids, where corticosteroids will dampen the inflammatory response. In essence, what we have realized so far is that sepsis is characterized by simultaneous hyperinflammation, which can cause and lead to lethal organ dysfunction, as well as immune paralysis, which does lead to various debilitating conditions, for example, viral reactivations and failure of tissue recovery. So altogether, this beckons the question, how does an aging immune system influence immune paralysis in sepsis? Can we at some point intervene and rejuvenate innate immune cells to reverse immune paralysis? A key seminal paper, a study by Peter Brendon in the US, looked at twins, old monozygotic twins, and also young monozygotic twins. And what these authors found was that the genetic makeup does not influence or partly influences the variability in the immune response. What does influence the immune response or variability therein were adaptive changes that are induced due to non-heritable factors. And these are shaping our immune system as we speak. And these factors are lifestyle factors or medications, vaccinations. So the environment here is a key determinant of how our immune system is going to react. And this whole field of study is known as epigenetics. And this is where we study chemical modifications to our DNA that are a result of environmental cues. Think of it as a switch, where more methylation on one end turns off a gene, whereas less methylation turns it on. And this, in itself, conveys identity to all our different cells. For example, a macrophage now knows that it is a macrophage, an immune cell, or an epithelial cell, a neuron. But what happens if this switch is malfunctional? What we're observing is that these cells somehow lose their identity, and this is the current hypothesis, what we're calling cell amnesia. In a recent study, we've recruited volunteers, participants, from the ages of 40 up until 92, obtained blood samples. We purified a very specific cell type, the monocyte, a key determinant of the immune response. And we can now measure these methylation or acetylation, all these chemical modifications that are happening to these cells, using next generation sequencing, bioinformatics, and now we're also leveraging on the tools of artificial intelligence and machine learning. What we've seen was that at various positions in our chromosomes, the percent methylation is decreasing by time. So we have demethylation with age. And this is in line with recent evidence in mice and other mammals where the loss of epigenetic information is a cause of mammalian aging. To further investigate this, together with my previous group in the Netherlands, we've manipulated a mouse, creating a mouse model that has a knockout for a particular gene, an enzyme, that removes these methylation marks. So this is a demethylase. When we observed these mice and challenged these mice in LPS endotoxemia, we found that mice lacking this enzyme had a significantly increased inflammatory response. So this was the cue that told us we're on the right direction, that indeed manipulating the epigenetic landscape of cells and also of whole systems, such as a mouse model, we can actually fine-tune the inflammatory response. So where does that leave us? The current understanding is that a cell gains its identity by flowing down what is called the Waddington Hills. And the Waddington Hills tell us that by rolling down this epigenetic landscape, a cell gains a particular identity until it falls into its particular spot as either a macrophage, which has very distinct functional states, or a T cell with its own functional states. But what happens during aging? What we do know is that this process is somewhat skewed, either towards one end of a cellular state, which is not required at the time, hence losing their identity. And we know of certain medications, interventions, that we can use to reverse or even improve on the cellular identity. So using these modifying interventions. Eventually, what we're looking for is an intervention or a combination of interventions that can rejuvenate our immune cells by functional reprogramming. I thank you all for your attention. I acknowledge all the people who have worked on this story so far, including my previous lab in Amsterdam and my current lab in Malta and Mater Dei. Thank you very much.